And we'll be starting off the afternoon session with a discussion of TAZ-CAMTA1 targeting. And our first speaker this afternoon will be Dr. Pobbati.
All right. So everybody able to hear me, see my pointer, I believe?
Yes, on both fronts. You're coming through
Cool.
great.
All right. So I'm Ajaybabu Pobbati, and I'm a project staff in Rubin's lab at the Cleveland Clinic. I'm also an assistant professor of molecular medicine at Cleveland Clinic Lerner College of Medicine. So the title of my talk is Evaluating Small Molecule Protein-Protein Interaction Inhibitors, or PPI inhibitors, for targeting EHE. So I thank the organizers for organizing this great meeting. And this is also the only meeting that I know of where you can just say EHE and everybody will understand what we are talking about without expanding fully what it is. Just a brief introduction to put things into perspective. We've seen this slide and Dr. Rubin explained it nicely in the initial thing. EHE, I like to reiterate, is a monogenic disease caused by 1,3 chromosomal translocation that produces the TAZ-CAMTA1 gene. And we in our lab have this great model system whereby expressing TAZ-CAMTA1 from the endogenous TAZ locus, we can recapitulate the human disease, and this is largely work by Caleb CV, who just talked prior to me, and also like Dr. Rubin explained, the TAZ-CAMTA1, the business part is it's through its interaction with TEAD, that's how it transforms the cells. So the TEAD-TC interaction is important because when you mutate this through disrupt this TC-TEAD interaction, TAZ-CAMTA1 is no longer transformative. So what do we get this from this? This is something that we don't get in many other cancers. We understand this cancer a lot more mechanistically. What is causing it? We know that TAZ-CAMTA1 is the driver. And to prevent its activity, we need to prevent its interaction with TEAD. So therefore, it makes a lot of sense to design a PPI inhibitor or a protein-protein interaction inhibitor to disrupt the TC-TEAD complex. Is this the only way to go? Believe me, this is the slide that I just put in after listening to Dr. Rubin's talk. He nicely mentioned that, that you can attack TAZ-CAMTA1 in this case because of the similarities in gene expression between TC, YAP, and TAZ. Whatever applicable for YAP, TAZ is also applicable for TC. You can target them upstream of TC. This is what I believe Ryan is going to talk about. Or you can target them directly, the TC-TEAD interaction. And you can also target the downstream components, in our case, like Dr. Rubin mentioned, the CTGF. So these three ways you can target the TAZ-CAMTA1. But what I'm going to primarily talk about is the tank approach, which is the group 2, directly targeting the TC-TEAD interaction. For the purpose of this talk, this is a TAZ-TEAD complex. You can also say it as a TAZ-CAMTA1 TEAD complex. So basically, the complex is, the way it works is that the TAZ-CAMTA1 interacts with TEAD by forming these interfaces. So basically, you can design a small molecule that either binds at the interface 2 pocket, or it can either bind at the interface 3 pocket as shown here. And these are the pockets that are on the surface. And there is an additional pocket that I found that which is a central pocket. So there are two broad ways of targeting the interaction between TC and TEAD, is either going through the surface approach are going through the central pocket approach. And both of them have their own advantages and disadvantages. The surface pocket approach is pretty straightforward and as a simple mechanism of action. That's because this is where TAZ-CAMTA1 is interacting. You're putting a small molecule there and prevents its interaction. This is a surface interaction, straightforward mechanism of action. But the disadvantage is that the pockets are shallow. So you never know that whether you can design a small molecule that is potent enough to disrupt this nanomolar affinity interaction. And the advantages and the disadvantages flip when you talk about central pocket. The central pocket is nice, it's druggable, but the disadvantage is that it has a complicated MOA, meaning that this is not the place where the TAZ-CAMTA1 binding. So if you want to disrupt this interaction, you want to go in an allosteric way. So there is a small molecule that has to bind here and allosterically disrupt TC-TEAD interaction. And that makes it a complicated MOA. It is further complicated by the fact that this central pocket is occupied by a ligand, which is a lipid, and you have to displace that ligand for your compound to be active. So you can go through a surface route or a central pocket route. And there is no right or wrong, but we decided to go through the surface route because it has a straightforward MOA. So this is largely work of Kepang Che, who initiated this screen, basically because the similarity between TAZ and TEAD complex, she used the TEAD and TAZ purified proteins. The reason for not using TAZ-CAMTA1 is TAZ-CAMTA1 is so difficult to purify. For that matter, even full-length TEAD and full-length TAZ are very difficult to purify. Kepang was resilient to purify this protein and come up with an assay. Basically, this assay, what happens is that when TAZ interacts with TEAD, you see an increase in the signal. And Kepang screened about 50,000 compounds and got 287 compounds as a hits. By the way, this paper is right now under revision. So these are still unpublished work. So when we looked at these hits and run a counter screen, basically what a counter screen does is that it checks whether the compounds that we identify are real PPI inhibitors or whether they are assay interfering compounds. And the way to do it is link the his and flag tag artificially and see if the compounds have an inhibition. Any compound that inhibits a counter screen is an assay interfering compound. Unfortunately, most of the compounds that we identify are assay interfering compounds, but nevertheless, we found this one molecule which looked like a real PPI inhibitor of the TAZ-TEAD complex. That's because it doesn't have an effect in the counter screen, but it only has an effect in the TAZ-TEAD alpha lysis screen. And now I'm like a mechanistic guy. I want to understand where these compounds bind. So I wanted to check whether this compound is binding to the central pocket or it's binding to the surface. The way I did it is that through use of a TAZ-TEAD FP assay. So FP stands for fluorescence polarization. So what it gives is that it reports the complex formation through a fluorescence polarization signal. So when TAZ is in complex with TEAD, you get a particular fluorescent polarization signal. When it is not in a complex with TEAD, you get another particular fluorescent polarization signal. So basically, I've used acylated and deacylated TEAD in my assay to understand whether the compound ATA is active. Given that ATA is active, when I used both forms of TEAD, suggests that the compound ATA is acting on the surface. The reason is when I use acylated TEAD or lipidated TEAD, it is blocking the central pocket. If the molecule cannot access the central pocket and still is active, suggests that the compound is binding on the surface. Now go back, where does it bind to the surface? Like briefly I said, it can either bind to this interface 2 region or it can bind to the interface 3 region. From this assay, which I'm calling this the VGLL1 TEAD FP assay, we draw conclusions that the compound ATA is binding to this place, which is the interface 3 pocket. And why do I say that? I want to draw your attention to a couple of works that I did a few years ago, where I showed that there is a protein called as VGLL1, which binds to TEAD, but only forms interface 2 and not 3. So I leverage this to show, because if any compound that inhibits VGLL1 TEAD FP assay is an interface 2 binder. And when you look at here, ATA does not interfere with a VGLL1 TEAD assay, suggesting that it acts as it binds to the TEAD on the surface at interface 3 region. My positive control that I used for this assay is a known interface 2 binder, which is the peptide 7, which is also identified through our work previously. This is mostly Elaine's work. So this gives a nice reduction signal in the VGLL1 TEAD assay, meaning that interface 2 binders work in this assay, whereas the interface 3 binders do not. Again, for a positive control for the interface 3, we used peptide 17, which is a known thing that binds to the interface 3. And both ATA and peptide 17 display similar profiles. Now, there is a problem with ATA when you put this molecule to medicinal chemists, they would say immediately, hey, they are 3 carboxylic acids, and it's going to be difficult to work with a molecule like that. And we did a lot of analog synthesis, and we still don't understand what is the group, functional group that we have to remove in order to make ATA more drug-like. An additional problem with ATA is that it also acts as PPI inhibitors of other complex. From the literature, you know that it acts as a PPI inhibitor of VWF GB1B complex and also tweak FN14 complex. So basically, you're having a selectivity issue here, meaning how can you modify ATA such that it only interrupts TAZ-CAMT complex as opposed to the other protein-protein interaction complex. So we have an uphill task here. So go back to the drawing board, start another screen, because we don't know exactly how to move forward with this. So briefly, the assay that I described before, the FP assay, is the same assay we used this as a screen. We use the YAP-TFE3 assay simply because it has a greater signal over background ratio. And you can see this is a saturation binding curve where you titrate the TEAD and you see an increase in the FP signal. So such FP assays are really good for doing a high throughput screen, and it works because the known peptide inhibitors, the peptide 7 and peptide 17, acts nicely in this FP assay. So we completed the screen. This is our unpublished results. So we again screened a library of about 58,240 compounds. This is a different library from what Kepang screened, and we got 71 hits that reduced the FP signal by greater than 50%. The compound that we used were the concentration of 25 micromolar. So this gives us a hit rate of 0.12%. I like to remind you that getting a PPI inhibitor is not easy, but we will get there. And a hit rate of 0.1% is kind of reasonable, because even before we started this screen, we anticipated that a hit rate is going to be about 0.2%, but we are pretty much right there. So we classified whatever the hits that we got is about into several clusters. So we have like 5 different clusters, and one of the cluster that frequently occur is, I'm just calling it as cluster A, because of the 71 hits, 10 compounds belong to cluster A. So again, coming to the point where, hey, does it bind to the central pocket or does it bind to the surface? Again, we are kind of more interested to get a surface binder. Again, when I used an acylated or deacylated TEAD, in this case, a palmitoylated or depalmitoylated TEAD, it's still active, suggesting that the compound that we identify, or a cluster A compound that I call it, is acting on the surface. So where is it acting? So let me remind you, when it is active in a VGLL1 TEAD assay, that means it is binding to the interface 2, and that's what we got. So we are lucky in the initial screen that we got something that is bind to interface 3. We have difficulty following that up. Now we're excited to identify something that binds to the interface 2. Are we going to follow up on this and try to see whether we can develop a drug-like molecule from this? So overall, I think I'm pretty much reaching my 10-minute time. So the take-home message that we want to convey is that PPI inhibitors of TAZ-TEAD interaction is actually a viable option, even for EHE treatment in particular. And you can develop a PPI inhibitor that targets either the TEAD surface or the central pocket. We have identified some surface PPI inhibitors that we are following up. They seem to bind to interface 2 and interface 3 pockets. And if you look at the literature, those surface binders are a little bit difficult to get. So we are a little bit excited that we got something like this. In the end, I acknowledge this would not have been possible if it had not been a team effort. The funding sources, thanks a lot for EHE Foundation for supporting our research. And I guess I'm done. Thanks for your attention.
Excellent. Thank you very much. Our next speaker will be Dr. Kanai.
Ajay, if you could unshare your presentation. Oh, thank you. All righty. All righty, so can everybody see my screen and pointer all right?
Looking great.
All right, thank you very much. So I would just like to start off by thanking the organizers of EHE 360 for inviting me to speak today. And without further ado, I'd like to talk to you about my project, which is inhibiting the tumorigenic function of TAZ-CAMTA1. So, of course, I think we all know a fair bit about EHE at this point, and I just want to address a couple quick points background points regarding EHE that are kind of pertinent to my project. The first being that EHE patients have pretty limited treatment options available to them, particularly in aggressive cases of EHE. And as work from many attendees has previously shown, about 90% of cases express a TAZ-CAMTA1 fusion protein, seen below here, while the other 10% express a YAP-TFE3 fusion protein. And as work from Caleb that was previously presented demonstrated in the genetically engineered mouse model of EHE, TAZ-CAMTA is sufficient to drive EHE formation, making it the disease-driving mutation. And other work from Jordan Driscoll in DJ Pan's lab demonstrated in a doxycycline off transgenic mouse model that loss of TAZ-CAMTA expression in tumors actually led to tumor regression. And so based on these findings, we surmise that either directly or indirectly targeting TAZ-CAMTA would likely be an effective treatment strategy for EHE. And so, as Ajay talked about, he's focused kind of on directly targeting the TAZ-CAMTA-TEAD interaction. But for us, gap in knowledge that was more interesting, or maybe just different, was that little is known about how TAZ-CAMTA is being regulated in cells. So we hypothesized that if we could identify pathways or genes upstream of TAZ-CAMTA1 that can repress TAZ-CAMTA1's functions, we might be able to therapeutically target those pathways to inhibit EHE growth and progression. So with that in mind, when we first started this project back in roughly 2019, the only models that were available for studying TAZ-CAMTA1 were NIH3T3 and HEK293 systems. And so we wanted to start off by determining whether TAZ-CAMTA1 TEAD transcriptional activity would induce activity in a YAP-TAZ TEAD responsive firefly luciferase transcriptional reporter plasmid. And so what we did was we took NIH3T3 cells and used retroviral constructs provided to us by Brian Rubin to infect NIH3T3 cells with increasing titers of retrovirus for expressing TAZ-CAMTA1. And as you can see, there was a dose-dependent increase in the transcriptional activity with the increasing doses of the retrovirus. And so similarly, we saw a dose-dependent increase in TAZ-CAMTA1 protein expression, suggesting that the system was working just fine for assaying TAZ-CAMTA1's transcriptional activity. Panel C, we're essentially just validating at the 100% dose that there was a significant difference relative to empty vector, which we chose the 100% dose due to the highest induction of this reporter. And essentially, we're hoping that that will give us the greatest dynamic range for identifying things that could repress TAZ-CAMTA transcriptional activity. And in panel D, we essentially repeated this with HEK293 cells, except we included a WW deletion mutant as well as an S51A mutant of TAZ-CAMTA1. The S51A mutant, as Brian and Ajay previously talked about, was for us just a proof of concept to make sure that our systems were working correctly. And for the WW deletion mutant, when patients have the TAZ-CAMTA1 fusion event, not all patients actually have a full-length WW domain. Sometimes it winds up being truncated due to the translocation. And so we wanted to make sure that any regulators that we identify that are able to repress TAZ-CAMTA would benefit patients both with the wild type full-length TAZ-CAMTA as well as the truncated version with the WW domain truncation. So with that system to study TAZ-CAMTA1 transcriptional activity, we then wanted to be able to study its tumorigenic function. And so we took the NIH3T3 systems and established a foci formation assay to as an in vitro readout for TAZ-CAMTA1's tumorigenic function. And we also established a subcutaneous tumorigenesis assay to have an in vivo readout for TAZ-CAMTA1's tumorigenic function. So now that we had systems to assay TAZ-CAMTA1's transcriptional activity and tumorigenic functions, we wanted to work towards identifying regulators of TAZ-CAMTA1. And given that our lab is a YAP-TAZ lab, we were drawn towards the TAZ portion of the fusion protein, given that it has two very important domains, as well as a variety of regulatory residues that are conserved in TAZ-CAMTA1 that might subject TAZ-CAMTA to regulatory pathways that normally act on TAZ. So with that in mind, we went through the literature and identified pathways that are known to regulate TAZ through that portion of the protein. And we used our 3T3 transcriptional reporter assay to identify things that might be able to repress TAZ-CAMTA's transcriptional activity. we identified a subset of genes that were able to. And so we went on to rescreen those same hits in the HEK293 models in both the wild type TAZ-CAMTA as well as the TAZ-CAMTA with the WW deletion mutant systems. And so as we can see here, all of those hits did continue to repress TAZ-CAMTA1's transcriptional activity in both models. And in the 293 system, it brought it back down to nearly empty vector levels of induction of the reporter assay, which was very promising. At the same time, we also just happened to have one-off regulator that looked kind of interesting. I noticed that there were the IQ domains in the carboxy terminus of TAZ-CAMTA1. And the IQ domains are actually putative calmodulin binding domains, and calmodulin, of course, being the calcium sensing protein in cells. And so I wondered whether calmodulin might be able to regulate TAZ-CAMTA1's transcriptional activity. So we took the same system, but the HEK293 system, and transfected in human calmodulin, and found that indeed we did actually see significant repression of TAZ-CAMTA's transcriptional activity, actually back to empty vector levels or baseline levels of reporter activity. So this was very promising, and we actually were able to write a grant through the EHE Foundation that was funded to pursue this as a regulator of TAZ-CAMTA1. And I'll thank them a little bit later on in my talk. So with all that being said, we have now identified several, candidate regulators of TAZ-CAMTA1. So moving forward, we're going to hope to determine if those regulators can also inhibit TAZ-CAMTA's tumorigenic function, not just its transcriptional functions. We're going to perform mechanistic studies, of course, to determine how those regulators are actually influencing TAZ-CAMTA1 functions. And then we would also like to determine if our regulators can inhibit TAZ-CAMTA in EHE cell lines, which brings me to the next part of my talk. As Caleb presented a little while ago, he talked about his genetically engineered mouse model of EHE that he was published in 2021 in Genes in Development. And as he mentioned, he was able to successfully isolate EHE mouse cell lines in vitro for use, which he graciously shared with us. And so for our studies, we initially wanted to focus on a couple things. One was optimizing cell culture conditions based on some of the initial information that Caleb provided us with, to make sure we could handle them, comfortably in a dish. And then we want to make sure that these cell lines could be infected with retroviruses and lentiviruses, which would allow us to pursue our candidate regulators in EHE cells. So the data is not shown here, but we were actually able to successfully infect these cells with both retros and lentiviruses. So with these foundations set up in place, we decided to focus then on model development for studying EHE using these cell lines. And so the first thing we actually tried was an ultrasound-guided intracardiac injection model. And so what this is, essentially we use ultrasound to guide a needle into the left ventricle of the mouse heart. And then we're able to inject labeled tumor cells into the heart. This model disseminates cells throughout the entire body of the mouse. But importantly for EHE specifically, this model does a really good job of delivering tumor cells to the liver, lung, and bone, 3 locations where EHE tumors generally form. And so we use this model with the mouse EHE cell line transduced with ZS green and injected those into mice hearts. So very recently, we actually went and wanted to euthanize 2 mice at an early time point to see what's happening in our mice. And so in both models, we found that we had some microtumors forming in both the liver and the lungs. And so, unfortunately, it's hard to see single cells or small tumors in the bones, even labeled with ZS green fluorophore. But we're hoping that at a later time point, there might be some tumors big enough that we can actually, have bone tumors forming from the injected cell lines. And so what's really nice about this model is not that it's just that it gives us an in vivo model for testing our regulators, but it also would allow us to determine if there are organ-specific differences in how our regulators are able to impact EHE growth. So if one regulator was able to grow, inhibit growth in the lung, but not the liver, this assay would allow us to tease that out. And so, for therapeutic testing and things moving forward, this would be kind of important, I think, for preclinical testing to determine which patients might most benefit from any treatments. And so one other interesting thing that happens with this model sometimes is that sometimes cells leak out of the heart, the left ventricle, and essentially are disseminated into the chest cavity, which we can usually visualize on the ultrasound. And so one of the 2 mice that we euthanized did look to have look like it had some cells leak out of the heart. So we included it as one of the early 2 time point mice to be euthanized. And so we actually found these reasonably large and well formed tumors in the chest cavity wall of that mouse. And so It's not clear if these are growing on the pleural surface or the pleural tissue. We haven't really done any sectioning or staining yet, but just by grossly, it looks like they might be growing in the pleura. And so, of course, we need to investigate this further, but this might actually be an interesting way of studying pleural tumors as opposed to parenchymal tumors, which I think would be kind of interesting, especially given that the pleural tumors tend to be the more lethal tumors for patients with EHE in the lungs. So this is just kind of an interesting observation, and we're hoping to pursue this a bit further. So future directions for this section, of course, we want to develop some in vitro assays for using the mouse EHE cell lines to validate our regulators a bit further. We want to confirm the TAZ-CAMTA dependence of the in vitro and in vivo models we established with the EHE cell lines, which Jordan Driscoll, I believe in the chat, asked a question earlier about that. That is something that we're also looking to do. We're working to do in parallel with Caleb. And so we're also looking to develop EHE cell line models, or rather use those models to further test our TAZ-CAMTA regulators and make sure that they can repress TAZ-CAMTA1 driven functions. So I just want to thank a whole lot of people, of course, my lab members, my PI, John Lamar, Janine Warren and Emily Norton, who are also PhD students in our lab, our collaborators at the Cleveland Clinic, Brian Rubin, Caleb CV, Ajaybabu Pobbati. And of course, I want to thank the funding agencies for this research. So for our initial foray into EHE research, we're funded by the EHE UK, USA, and Australia branches. And with this new CalModulant grant, we are being funded by the USA and Australian branches. So of course, thank you so much for the funding for our projects. And of course, I want to thank the Cleveland Clinic's EHE Research Collaborative through the Cleveland Clinic, which is funded by the Margie and Robert E. Peterson Foundations. And lastly, I want to thank Guy Weinberg and the Crab Foundation and Eaton Foundations for inviting me to the annual Telluride conference for EHE, and of course helping sponsor some of my travel there. So that's all I have.
Excellent. I'll invite Dr. Pobbati back for a Q&A, small group here. And Ryan, if you'd like to stay around as well, we'll have a small panel discussion here. And we do have a question that's come in from the audience for Dr. Pobbati. Question is, do you know if the TEAD in the complex with the TAZ-CAMTA1 is palmitoylated? And then follow-on question, will palmitylation status affect binding of some of the PPIs?
So when we did a lot of in vitro studies using a palmitoylated and a non-palmitoylated version of TEAD, and we don't see any difference. At least in vitro, the palmitylation status doesn't affect anything. And this kind of reflects at least in vitro studies from other groups like Patrick Chen's group. But in vivo, it's a difficult story. We don't know how this is happening. And it's also confusing mechanistically how a palmitylation is able to allosterically modify it in cells. So at least in vitro, we see no effect, whether TEAD is palmitoylated or not palmitoylated. But in cells, it may have an effect, although the mechanism is unclear.
Excellent, thank you. And then, with your second screen, you have the cluster A with the 10 compounds and some other clusters. And I know it's too preliminary to get into any details about any of those at this point because you have a lot of experimenting to do. But in looking at some of the structures of the compounds within the hits in the second screen, can you say anything about whether some of them structurally look like they're good drug candidates?
They are. I mean, that's why we are following up on these. I mean, we have a whole medicinal chemistry team working on it at the Cleveland Clinic Therapeutics Discovery team. We will only follow up if it looks like drug-like candidates. So this is where a collaboration with the medicinal chemists and the biologists have to merge, because a lot of time we don't know if this compound could be translated into a drug candidate, and this is where they play a critical role in saying, look, I mean, this will never be some stuff like that.
Yeah, kind of a similar question for Ryan's. It looks like in your initial screens you found quite a few upstream pathways that might be interesting regulators. Is there anything you can say at this point, and I know it's early, but in terms of whether any of those pathways have existing drugs that might be able to be used to regulate them rather than requiring discovery of novel compounds?
Yeah, so that's actually kind of how we decided which regulators to pursue more heavily right off the bat. And so In particular, we're looking at the AMP kinases as well as GSK3 beta. So in AMP kinase, there are actually clinically used drugs like metformin that are already in place that would be act on activating AMP kinase. And similarly, it's also acting, AMP kinase can also act through the mTOR pathway by inhibiting mTOR. So this might be something that's either acting similarly to how the mTOR inhibitors might be acting, or could be another way, another strategy that could be acting complementarily to like sirolimus or other mTOR inhibitors. And GSK3 beta, I believe, there is inhibited by the AKT pathway, and so there are some AKT inhibitors that are in trial, I believe, phase 2 at the moment, or something along those lines. So those might be other possibilities that could be repurposed quickly for EHE treatment if things worked out.
Excellent, thank you. And one of the other advantages of your approach is when you're identifying pathways is, in theory, if you have multiple hits in multiple pathways, you could have two compounds, right? Potentially having an additive or even a synergistic effect. I know the sort of the regulatory side of that becomes a bit more complex, but have you been considering or looking at combinations at all?
Yeah, so we're initially just trying to make sure we have validated targets that function to inhibit EHE growth, especially in these new models that we're starting to develop. And so once we have those in place, that would certainly be one of the future directions to test available compounds and possibly do combination therapies, either with existing ones like sirolimus and or trametinib, or with new compounds based on our regulator screens. So.
Thank you. And we just had a new question come in from the audience. Your transcriptional reporter assay for TAZ-CAMTA1 seems to vary widely. You show greater than 60-fold induction in some experiment, but in the CAMK experiment, a baseline stimulation was only 2 to 3-fold. Could CAMK suppress a higher baseline?
Yeah, so actually what we found is some research from, I believe, Dr. Valery Kuskov has also shown that over time, TAZ-CAMTA expression diminishes even with a DOX-inducible system. And so similarly, we've actually found that over time, cells seem to, of their own accord, inhibit some of the induction of our transcriptional reporter assay, which we're still looking into whether that's through, an expression level change, like an inhibition of the TAZ-CAMTA1 protein expression, or maybe just basal endogenous pathways are managing to inhibit the TAZ-CAMTA1's transcriptional activity. So we don't think that that's necessarily just through the regulators. So we've tried at different time points for our regulators, and the fold changes seem relatively consistent across our regulators.
Got it. Now, this last question that has shown up from the audience, I think would apply to both of you. It might be a challenging one to answer. In all the model work, we are seeing progress made in tumor generation and considering how we can treat progressive EHE. Are we seeing anything that begins to explain why some EHE is indolent? And I know that's a big general question, but turning that to some of the approaches you're looking at and some of the model discussions, is there anything you can say about how this research might inform how to address indolent?
Ajay?
We can only do like a hand waving thing here, just like you pointed out in there. So one hypothesis that we have, that the progression from an indolent to an aggressive phenotype could be through the through the tumor acquiring additional secondary mutations. So one possibility could be through, like what Caleb mentioned, through whether you get an additional hit of CDKN2 loss that could convert them from being indolent to aggressive. But that's a hypothesis we need to test thoroughly, but it looks like an attractive hypothesis. Like the accruement of secondary mutations could be the transition that makes it from being an indolent to an aggressive phenotype.
Excellent. And that wraps up the questions we've received. Thank you both so much for your time, and hope you stick around and enjoy the rest of the conference. And we will now move on to the second part of this session, talking about gene fusions in EHE, and our first speaker will be Keith Garcia. And Keith, we're seeing your slides, but you're on mute. There you go.
All right, so hello everyone. I'm Keith Garcia. I'm a 5th-year PhD candidate in the TANAS lab at the University of Iowa. And today I'll be talking about how the TAZ-CAMTA1 and YAP-TFE3 gene fusions can alter the TAZ and YAP transcriptional program by interacting with the ATAC complex. So a little background on EHE, it's a malignant vascular neoplasm that can essentially arise anywhere in the body, often presenting as multifocal or metastatic lesions in the lungs or liver early in its clinical course. And it was originally believed that to affect men and women equally, but emerging evidence is starting to suggest that perhaps females may be more susceptible to this disease. And interestingly, under the microscope, when you look at the H&E section of EHE, one interesting characteristic of this tumor is that they form these cord-like structures embedded in this myxohyaline matrix. So in addition to EHC, our lab studies the Hippo tumor suppressor pathway, in which the end effectors are the transcriptional coactivators, TAZ and YAP. Interestingly enough, in about 85% of EHE cases, they contain this TAZ-CAMTA1 gene fusion, whereas the remaining 15% or so cases contain this YAP-TFE3 gene fusion. And so structurally, when you look at the domain architecture below here, at the end terminus of TAZ-CAMTA1 and YAP-TFE3 is that they retain their TEAD binding domain, which is fusion framed to the transcription factors CAMTA1 and TFE3 respectively, which provide additional interacting domains. And much of the rigorous mechanistic work regarding these gene fusions has really focused on the N-terminal portion of these fusions. And as Frank will potentially talk about after me and has alluded to in his papers, that at least for YAP-TFE3, the C-terminal component does have a functional role. And so the focus of my talk really will begin to dissect, how are the C-terminal fusion partners of these gene fusions contributing to the oncogenic phenotype of these two oncogenic proteins. And so many of us here that study these gene fusions can appreciate the fact that in many different cell lines, expression of these fusions is able to drive cellular transformation. And so for example, in mouse 3T3 cells, as well as a mildly oncogenic liposarcoma cell line, ectopic expression of these fusions is able to drive anchorage-independent growth under low attachment conditions relative to the empty vector controls. And further expanding these in vitro observations into a mouse xenograft model, as you can see, expression of these fusions can initiate tumorigenesis early on relative to the empty vector control. And now there are two genetically engineered mouse models of EHE that have been independently developed by Dr. Driscoll and Dr. Seavey. And really the take-home message with this slide is, and their work is simply expressing the WWTR1 CAMTA1 transgene is sufficient enough to initiate tumorigenesis and drive EHE formation. And so next, focusing more on YAP-TFE3, we're interested in assessing, which components of YAP-TFE3 are critical for its oncogenic capacity. And so the test is we've generated truncation mutants containing the N-terminal and C-terminal portions identified as YAP trunk or TFE3 trunk, and stably express them in YAP-TFE3 cells. So shown here on the right, you can see that full-length YAP-TFE3 can form these nice colonies in soft agar, whereas the truncation mutants shown below here fail to do so, providing evidence that you need both components of the N and C terminus to fully transform cells in vitro. And analogous to TAZ-CAMTA1, we recognize that YAP-TFE3 retains its TEAD binding domain at its N terminus. And so we hypothesize that it functions through the TEAD transcription factors to drive cellular transformation in vitro. And so in 3T3 cells that stably expressed YAP-TFE3, we subsequently knock down TEAD1 utilizing two independent short hairpin RNAs. So as you can see here on the right, when you knock down and disrupt the TEADs in these cells, it significantly abrogates YAP-TFE3's ability to drive anchorage-independent growth. And so like TAZ-CAMTA1, which has previously been shown to rely on TEAD transcription factors, YAP-TFE3 relies on TEAD1, and these fusions simply rely on these transcription factors to mediate their oncogenic function. And it's because of that fact that we suspect and wondered whether they simply recapitulate a wild-type TAZ and YAP transcriptional program. So we performed bulk RNA-seq analysis, and what we observed was that the transcriptomes for YAP-TFE3 and TAZ-CAMTA1 significantly overlapped with full-length YAP and TAZ, respectively. But despite this considerable overlap in the transcriptomes, YAP-TFE3 as well as TAZ-CAMTA1 can derive unique target gene expression. And we suspected that this perhaps could be due to altered binding properties not observed in full-length TAZ and YAP. And to begin to test that, we subsequently performed ChIP-seq analysis, and analogous to our RNA-seq data, essentially what we observed was that the chromatin binding profiles for TAZ-CAMTA1 and YAP-TFE3 did overlap considerably with full-length TAZ and YAP respectively. But again, despite this overlap in the chromatin binding, both TAZ-CAMTA1 and YAP-TFE3 can bind to unique sets of genes. And this can be explained in part when we perform motif enrichment analysis, which shows that the fusions do occupy regions with the T motifs. But in addition to that, we observed an enrichment for this EGR2 motif and MITF motif in the TAZ-CAMTA1 and YAP-TFE3 ChIP-seq datasets. And these two motifs are actually primarily enriched in the wild-type CAMTA1 and TFE3 ChIP-seq datasets. And so this data provides evidence that the C termini of these gene fusions have altered DNA binding properties, not observed in full-length TAZ and YAP. And so to begin to address, how these global transcriptomic and chromatin binding changes may be occurring, we next wanted to address whether these fusions can further augment the TAZ and YAP transcriptional program by modulating the chromatin landscape. And to begin to test this, we performed ATAC-seq analysis, which basically assesses, how accessible the DNA is for further transcriptional regulation, or how open is the genome. And so showing these tables to the left, you can see that TAZ-CAMTA1 and YAP-TFE3 are able to stimulate an increase in the number of accessible genes that are available for potential transcriptional regulation when compared to full-length TAZ and YAP. And strikingly, when we performed principal component analysis, the overall euchromatin landscape of these fusions largely resembles that of wild-type TFE3 and CAMTA1, providing additional evidence that the C termini of these gene fusions really play an important role in further shaping the chromatin landscape to potentially drive this unique transcriptional program. And because we've had some understanding that wild-type TAZ and YAP are able to interact with epigenetic cofactors such as members of the switch-SNF complex, we suspected that perhaps through the C-terminal fusion partners may be providing additional protein-protein interactions with epigenetic cofactors. So to test this, we performed a bio-ID mass spectrometry screen in collaboration with Anne Cloud Gingras at the University of Toronto. And visually, what stood out to us is that the interactomes for TAZ-CAMTA1 as well as YAP-TFE3 largely overlapped with their C-terminal fusion partners, CAMTA1 and TFE3 respectively, rather than YAP and TAZ. And many of these interactors are actually enriched for chromatin modifying cofactors involved in histone modifying complexes and ATP-dependent chromatin remodelers. And so next, we wanted to functionally characterize, well, which of these many interactors are important with respect to the oncogenic properties of these gene fusions. And so essentially, we've identified 18 top hits in our interactome, and we subsequently performed a targeted RNAi screen for these 18 genes/proteins, and really wanted to address whether if you disrupt these interactors, does that reduce and abrogate the fusion's ability to drive anchorage-independent growth? And so we utilized 5 SHs to test this, targeting these genes/proteins, and we rank ordered them. So the more SHs that prevented the fusion's ability to drive anchorage-independent growth, the more confident we were that these interactors were important in its function. And so briefly, the top hits that we observed in which all 5 SHs abrogated TAZ-CAMTA1's ability to drive anchorage-independent growth were these 2 proteins, YEATS2 and ZZZ3. And so these proteins are members of the ATAC histone acetyltransferase complex. And so just some background on this complex, it contains a HAT subunit containing GCN5 or PCAF that are mutually exclusive. YEATS2 is a scaffolding subunit, and it's also been recently identified as an oncoprotein in non-small cell lung cancer. And it's a reader protein, so it's able to bind and recognize acylated histone 3 lysine residues on histone tails. And ZZZ3, like YEATS2, is another reader module of this complex and can actually recognize unmodified histone 3 tails. And what was interesting to us is that CAT14 actually also fell out of our mass spec screen and preferentially interacted with TAZ-CAMTA1 and YAP-TFE3 than they did to TAZ and YAP. And this other component is primarily acts as an adapter and does have weak acetyltransferase activity. And so lastly, what we next wanted to begin to address was, well, how is ATAC mediating the transcriptional output of these gene fusions? And so we performed RNA-seq analysis in which we've knocked down either YEATS2 or ZZZ3 in cells that stably express TAZ-CAMTA1 and YAP-TFE3. And we performed principal component analysis. As you can see, when you disrupt members of the ATAC complex, it does cause a variance and change to the transcriptomes of TAZ-CAMTA1 and YAP-TFE3. But these changes are actually appear to be more drastic for YAP-TFE3 than they are for TAZ-CAMTA1 when you're comparing them to the non-targeting controls. And we suspect that this modest effect when you perturb ATAC with respect to the TAZ-CAMTA1 expressing cells could be due to the fact that TAZ-CAMTA1 is able to interact with many other chromatin regulators. And so we suspect that these other complexes may also be important in TAZ-CAMTA1's oncogenic activity. And so our working model that we have so far is that these fusions to their N-terminal T binding domain interact with the T transcription factors. The properties contributed by their C-terminal fusion partners are facilitating additional protein-protein interactions with epigenetic cofactors, such as members of the ATAC complex, to further augment the basal TAZ and YAP transcriptional program to drive oncogenesis. And so with that, I'd like to thank members of the TANAS lab, Dr. TANAS for his mentorship, our collaborators, Dr. Michael Chimenti that did a lot of analysis of our sequencing data, and Cloud that helped us with the mass spec, and Kieran Harvey, as well as the EHE Foundation, and specifically Guy Weinberg for really providing support for me to go to Telluride and present some of this science with respect to that Hippo pathway meeting. So thank you.
Thank you much. And our last speaker in this small group is Dr. Szulzewsky.
Thank you so much. Can you all see and hear me?
Yes.
See my screen? Okay. So first of all, thank you so much for the opportunity to speak here and present my work that I performed in the lab of Eric Holland at Edward Hutch. I'm going to talk about the different oncogenic functions of different YAP1 gene fusions. So YAP1 is a transcriptional co-activator that is controlled by the Hippo signaling pathway. And the Hippo pathway is a series of serine threonine kinases that ultimately phosphorylate YAP1 and also TAZ several serine residues and control its activation by both cytoplasmic retention and also proteasomal degradation. And activated YAP1 and also TAZ activity can be found in several cancers. However, this is rarely achieved by activating point mutations in the actual YAP1 sequence, but rather through inactivating mutations in the upstream Hippo signaling tumor suppressive pathway. However, nature found a way to actually generate activating mutations in YAP1, and they're the so-called YAP1 gene fusions. And several YAP1 gene fusions have been found in different cancers all over the body. And we, in our paper that we published in 2020, we looked at 4 of them: YAP1 mammary 1, YAP1 FAM found in epidemoma, YAP1 SS18, and also YAP1 TFE3 found in EHE. And I'm just going to briefly go over the different hallmarks that we came up with in our paper, that basically have unified functions of the different YAP1 gene fusions. And here on the left side, you can see a diagram of the structure of these gene fusions, and you can see that they all retain the very N-terminal part of YAP1, including the T binding domain, and they then lose or retain different parts of YAP1, and that is replaced or substituted by C-terminal fusion partners. And the hallmarks that we came up with, and I'm going to go into more detail in the following slides, but 1, all of these gene fusions that we looked at, they're oncogenic when we expressed them in mice. 2, these YAP1 gene fusions exert both YAP activity and also activity of the C-terminal fusion partner, and this is most prominent for YAP1 TFE3 that's found in EHE. Number 3, these YAP1 fusions are resistant to inhibitory Hippo pathway signaling, A, through nuclear localization, which is achieved by the presence of nuclear localization sequences in the C-terminal fusion partners, and B, through resistance to proteasomal degradation, which is achieved by the loss of the serine 397 residue in the YAP1 sequence that is lost in all of these YAP1 gene fusions. And number 4, we could show that these YAP1 fusions require TAZ for their oncogenic functions. Basically, this transcription factor that is necessary for the activity of wild-type YAP1 is also required for the activity of these YAP1 fusions. And we used our RKS-TVA system, which is a system for somatic cell gene transfer. And we were able to basically express these different fusions in nestin-positive cells in different locations in these mice. And these mice have also CDK2 null background. And here I'm showing data from hind limb injections or expression in the hind limb. And we can see that all 4 gene fusions that we analyzed are able to form tumors in these mice. And for TFE3, for YAP1 TFE3, we're not generating EHE And this is most likely because we're not hitting the right cell of origin for EHE. But it has to be said that none of the wild-type proteins cause cancer, however, all of these fusions do. Number two, we looked at the activity of these different fusions, and we performed different assays, and one of them is basically RNA sequencing. And we expressed the wild-type versions of these gene fusions, wild-type YAP1 or the wild-type CAMTA1 fusion partners, as well as 2SA, 2-point mutant YAP1, which is a constitutively active YAP1, or 3 of these gene fusions, including YAP1 TFE3. And you can see that all of these wild-type versions, they basically cluster very closely together with a GFP control. They don't elicit large gene expression changes. However, 2-point mutant YAP1, oncogenic YAP1 itself already basically clusters away from these wild-type versions. But then if you look at these YAP1 gene fusions, they furthermore cluster away from just 2-point mutant YAP1, showing that these fusions bring something in addition to just pure YAP1 signaling. And when we look at the gene expression changes activated by these fusions and we overlap them, we can see that there is an overlap between all of these 3 gene fusions, a large overlap. And when we compare that to the signaling from 2-point mutant YAP1, we can see that it's highly enriched, showing that what is similar between these gene fusions is YAP1 signaling. But then it's not just YAP1 signaling for these fusions. And we also did cut-and-run chip sequencing, and I'm showing data here for YAP1 TFE3. And we could show that we get a large number of peaks for YAP1 TFE3. And we also were able to get robust peaks for wild-type TFE3. And we can basically see that almost all of these peaks generated by wild-type TFE3 are also found in YAP1 TFE3, showing that YAP1 TFE3 basically retains most of the function of wild-type TFE3. But something that has to be said is that the number of peaks generated by wild-type TFE3 and YAP1 TFE3 is dramatically lower than for for wild-type YAP1 or wild-type TFE3. It's dramatically lower than the peaks we generated for YAP1 TFE3, showing that basically the fusion is not just the sum of both wild-type versions, but it's something in addition. And we also have a TFE3 responsive luciferase reporter assay in which we can show that both wild-type TFE3 and YAP1 TFE3 show activity. And when we inject these different fusions in the brain in our mouse model, and we also get tumors, and you can see that these fusions cause tumors that look very different among each other. And especially for YAP1 TFE3, these tumors that we generate, again, it's not EHE, but we see that the histomorphology of these tumors is very different compared to both non-fusion oncogenic YAP1 and also the different YAP1 fusions, showing that the function of YAP1 TFE3 is very different to these other fusions. And right now we're collaborating with the lab of Munitanas to basically look at like what do these different domains in the TFE3 sequence in YAP1 TFE3, how do they contribute to these functions and also the morphology of these tumors that we generate. The next thing that we looked at is how do these YAP1 fusions, how do they respond to Hippo signaling pathway, and to basically mimic this, we used a YAP1 responsive luciferase assay where we expressed either YAP1, wild-type YAP1 or the different fusions, and I'm showing here for YAP1 FAM or YAP1 S18. And we also co-expressed the 3 Hippo pathway tumor suppressor members, MST, MOP, and LATS, and we can see for wild-type YAP1, we see dramatic reduction in the activity of wild-type YAP1. However, for the fusions, we don't see this drop, showing that these fusions are much more resistant to Hippo signaling pathway. And we think that's in part due to constitutive localization of these fusions. We basically transfected HEK cells with these different constructs and then cultured them at different cell densities. And you can see here for wild-type YAP1 at high densities, it's excluded from the nucleus. However, all of these fusions, they basically are retained as stuck in the nucleus. And this is likely due to the function of the actual wild-type, C-terminal wild-type partners. And then in addition, we looked at can these fusions still be degraded, and we generated stable cell lines of 3D3 cells that are transduced with either wild-type YAP1 or these different fusions, and then culture them at low and high densities and perform Western blot to measure the abundance of the protein. And we can see that wild-type YAP1 is significantly reduced, the protein is significantly reduced, whereas none of these fusions, it's not true for any of these fusions. They're basically stable, so they cannot be regulated anymore. And the last part that we looked at is, do do these fusions still require TAZ for their oncogenic functions? And as I said before, all of these fusions, and there's additional fusions that we're looking at right now, and it's true for all of those, and they basically retain the TAZ binding domain. And we performed assays, again, luciferase assays, where we knocked down all TAZ 1 through 4 at once, and then looked at the luciferase assay in a YAP1 responsive promoter assay, and we see that we see significant reduction of this YAP activity for all of these fusions upon knockdown of these combined knockdown of all 4 TAZ. And when we do the reciprocal experiment, where instead of knocking down TAZ, we use a mutant version, S94A version, that cannot bind to TAZ anymore. And we do, again, RNA sequencing in these human neurostem cells that are stably transfected. And you see this graphic PCA plot that I showed before. And then when you overlap these mutants of YAP1 mammaly1 and YAP1 fem that cannot bind to TAZ anymore, you see that there's a dramatic shift in the activity, showing that the large part of the activity is basically relies on TAZ. It's not all of that, but a large part basically relies on the interaction with TAZ. And again, when we also take these mutant versions that cannot bind to TAZ and we inject them in our mouse model in mice, and we see either complete abrogation of tumor forming capacity, or we see significant reduction here for YAP1 TFE3. And the last thing that we did is that we took our mouse tumors and we generated tumor slices and we cultured them, and we can treat them with different drugs. And one drug that we use is verteporfin, which is a known inhibitor of YAP1 TAZ interaction. It's a very dirty drug, but it has been used before. And we also used other drugs for that, and we see like similar effects. And we see that verteporfin treatment, or basically YAP1 TAZ interaction inhibition, is able to reduce the viability of these slices with an increase in apoptosis. And right now, in a collaboration with Tara and Gujral, we're doing like screens of small molecule inhibitors to see what kind of kinase inhibition can be used to inhibit the viability of these tumor slices. And this is the end of my talk. And if you're interested, please check out my paper, or like our paper. And I would like to thank all of our collaborators, and again, the organizers of this conference, for inviting me to talk. Thank you so much.
Thank you as well. And I'll invite Keith back for a group Q&A session. All right, we actually have some one question for each of you from the attendees. Start off with a question for Keith. Is it possible to characterize your TC transcriptional profile into TEAD and EGR2 targets and interrogate which of these is more oncogenic?
Yeah, we haven't begun to analyze that and identify like maybe what regions TAZ-CAMTA1 is binding to, and what genes with respect to the C terminus that TC is binding to that may be oncogenic. We haven't really looked specifically at the gene level. Basically, what we have identified is that the C terminus can bind to unique regions, and these regions occupy the EGR2 motif, but whether those genes are oncogenic, we haven't explored that yet.
A question for Frank. Do you need CDKN2A null background to cause tumors? And you're on mute right now.
My apologies. We don't need that background to cause tumors, but it greatly enhances the size of these tumors and the oncogenicity. For YAP1 TFE3, we see small tumorlets in the brain in CDKN2A wild-type mice, but then when we use CDKN2A null mice, the tumor size is greatly increased.
Thank you much. And then sort of a challenging future directed question for each of you. I'll start with Keith. Understanding what we know from your data about the C-terminal interaction in the model you've produced, what can you say about it as a potential target for therapeutics in terms of disrupting its interaction with whatever it's interacting with in the cell?
Yeah, so targeting these epigenetic complexes is kind of like an emerging field. Like switch SNF is like mutated in 20% of cancers, and there's already a spin-off drug company that's targeting it. With respect to the ATAC complex, the enzymatic subunit GCN5 and PCAF are not unique to ATAC, so that's a challenging target. But what is unique are these reader modules, and I know there's work done at least in different leukemias, targeting the YATES domain, and that's another therapeutic opportunity to target in addition to the TAZ, right? Because if you could target TAZ, that's also a big important vulnerability for these fusions. So yeah, so the enzyme component of this complex may be difficult, but the reader models may be a nice avenue to pursue.
And then do you see any diversity among C-terminal sequences in terms of different fusions that are showing up, truncations that in any way influence or put into categories the types of results you're seeing?
Yeah, so for other TAZ and YAP gene fusions, many of the C-terminal fusion partners, like Frank was highlighting, many of them are transcription factors. So we suspect that, and they all retain this transactivating domain, which plays a role in recruiting different subunits of these epigenetic complexes. So we suspect that there may be overlap or there may be distinctions, but we feel like based off of that model diagram that it showed that they may be functioning through the TAZ at the N-terminus, and whatever C-terminal fusion partner, if it's a transcription factor, may be recruiting these epigenetic complexes.
Then the challenging sort of future-looking question for Frank, one of the targeting modalities people look at for potentially treating cancer, where you've got transcription factors or nuclear cytoplasmic location of proteins being an issue, is can you affect localization? And your data is showing localization is playing a role here. Is there anything you can say about the future direction in terms of being able to regulate the process as a potential future therapeutic direction?
We're actually not going in that direction. So we're literally just more looking downstream and what kind of kinases are activated, like secondary effects of these fusions. Basically, what kind of kinases are activated and can we target those? Like we see platelet PDGF receptor signaling going up in several of those, and we're testing different compounds to basically inhibit the viability of these tumor cells and also our tumor slices with those.
And related to mechanism, you mentioned you'd done the testing with the, let's see if I pronounce it right, the verterophrin drug, if I pronounce that. What can you say about the mechanism of how that drug works?
So, I mean, the verterophrin is a very dirty drug, right? And it's probably affecting tons of other things. I mean, the postulated thing is basically that it inhibits this interaction between TAZ and YAP1. And we're also testing other compounds, and we're collaborating with collaborators at UCSF that basically came up with a list of YAP1 TAZ and inhibitors, interaction inhibitors, and we're also working with Vivace to basically look at other inhibitors. And we're seeing promising results that show like similar ones. And one thing that it might be helpful also for the TAZ-CAMTA community, like one thing that we're doing right now is that we're using the DTAG system. I'm not sure if you've heard of that. It's basically tagging our fusion, and we're working for 5SA YAP1 with that. It's basically giving it an FKBP tag, and then you can express it as in vitro, like in our mouse model. And when you get when you give a little molecule, basically the fusion gets degraded, and you can establish like a mouse, like a tumor, and then you can add this drug. And we're doing this right now in slice culture, and you can degrade the actual oncogenic fusion, and then you can look at the downstream effects like after you degrade it. And we see similar effects between verterophrin and like the different YAP1 TAZ interaction inhibitors and this DTAG degradation that we see like in decrease of CTGF, SIR61. And right now we're basically looking at like what is the effect when you take away the fusion, or like 5SA YAP1, what does it do with the tumor cells?
Excellent. Well, thank you both so much for your time today. That addresses all the questions that have come into the queue. We'll end this part of the session and move on to our next. And we have one more technical presentation before our next break. Our next speaker will be Dr. Subramaniam.
Hi, can you hear me?
You're coming through good. You just switched to presenter mode, though. The previous mode would probably be the better way to view it.
I see. Okay.
I think you can do that from the display settings on the top center. Click on that, and there should be a swap to presenter view, the top option. If it'll let you do that.
Yeah, it would not let me do that. Let me. Oh.
Click that, and we should be in great shape. Looks perfect. Slides look good. Your audio is on mute.
Okay. Is this better?
Perfect on all accounts.
Okay, all right, thank you. Sorry for that technical difficulty. I thought I was supposed to be in presenter mode, my mistake. Good afternoon, everyone. I'm Aparna Subramaniam, and I am a graduate research assistant at the Sarcoma Medical Oncology Department at MD Anderson Cancer Center. I am also a PhD candidate at the UT Health Science Center, and my advisor is Dr. Vinod Ravi. Today, I will be presenting on the modulation of YAP and TAZ by statins in EHE and its association with survival outcomes in patients. I have no conflicts to disclose in relation to this presentation. This will be a very brief presentation going over the outline. There will be an introduction with the background of statins in EHE, the retrospective study of statins in EHE that we conducted, and we'll touch upon briefly on the future directions of research in this area. So as we know, epithelioid hemangioendothelioma is a rare vascular sarcoma of endothelial origin. Patients are usually asymptomatic, with initial diagnosis often being incidental. Most of the patients have metastatic disease at the time of diagnosis. EHE displays a variable clinical behavior ranging from indolent to highly aggressive disease. 90% of the cases have the pathognomonic TAZ-CAMTA1 fusion, as we have seen, and the YAP1 TFE3 fusion is less common and seen in approximately 10% of the cases. Surgical resection is the primary treatment for localized disease, and treatment for advanced or metastatic disease is undefined. Active surveillance in asymptomatic patients with a stable disease is an accepted treatment or management strategy in EHE. We now look at the Hippo pathway, which, as we have seen, is a pathway seen in the YAP-TAZ effectors. YAP-TAZ, as we've seen in the previous slide about the fusions, they are downstream effectors of the Hippo pathway. And the core of the Hippo signaling consists of a group of kinases, the MST1-2 and the LATS1-2, and the adapter proteins, the SAV1 and the MOB1. So when the Hippo signaling is on, as you see on the right panel, the kinases phosphorylate YAP-TAZ, and which results in the cytoplasmic retention of YAP and TAZ. In the cytoplasm, YAP and TAZ then undergo proteasomal degradation, and the VGLL4, which is in the nucleus, interacts with TEAD and represses transcription. Whereas when the Hippo pathway is in the off state, as you see in the left panel, there is no phosphorylation of YAP and TAZ, resulting in YAP and TAZ translocating to the nucleus, which then regulate the activity of several transcription factors, leading to expression of Hippo target genes that are involved in cancer development and progression. So Hippo signaling is a complex tumor suppressor, and its deregulation is a key feature in many cancers. We now look at the mevalonate pathway, which is an essential pathway that uses acetyl-CoA for the production of sterols and several isoprenoid metabolites that are essential for several biological processes. Recent studies have shown that the MVA pathway enzymes and their metabolites are essential for the survival of several cancer cell lines. So the mevalonate pathway and its metabolites are key dependency in tumor development. Looking at the interplay between the Hippo and the mevalonate pathway, we see that the mevalonate pathway metabolite GGPP, results in the activation of Rho-GTPase by prenylation and membrane association. Rho-GTPase then results in the activation of F-actin, and which suppresses LATS1 and 2 kinases in the Hippo pathway, leading to the YAP-TAZ nuclear localization and activation, leading to gene expression and tumor growth. This interaction between the Hippo and mevalonate pathway is essential in tumor development and can be a target for YAP-TAZ in human cancers. Based on this background of the Hippo and the mevalonate pathway, we look at the rationale for the study of statins in EHE. So when we look at statins, statins are HMG-CoA reductase inhibitors. HMG-CoA reductase is a rate-limiting enzyme in the mevalonate pathway that actually produces the GGPP. So once statins inhibit the metabolites of the mevalonate pathway, this results in the inhibition of YAP. It results in the inhibition of LATS1 and 2 in the kinase in the Hippo pathway, and does not allow the nuclear localization of YAP and TAZ. And as we have seen, TAZ-CAMTA fusion results in the nuclear localization and constitutive activation of TAZ in EHE, and statins decrease this nuclear localization. They have been shown to decrease growth and increase apoptosis in many cancer types, and statins as a class of drugs have been used for a long time to reduce cholesterol levels in patients with hypercholesterolemia by the inhibition of HMG-CoA. With this rationale in mind, we did a retrospective study of statins in EHE, and patients with the diagnosis of EHE were identified using an institutional electronic database. Patients were screened for statin use through their medical records. Demographic, clinical, and survival outcome data was abstracted in these patients, and the Kaplan-Meier method was used to assess overall survival for these patients. Findings from this study were also submitted as an abstract in 2020 at the ASCO meeting. We were able to identify a cohort of 225 patients, of which 27 patients had statin use recorded, and 198 patients had no statin use. We see that age of diagnosis was higher for the people in the statin group compared to the non-statin group. The population was predominantly white, and the liver was the most common site of primary tumor of EHE. Of the statins that were recorded, atorvastatin was the most common, with 14 patients having received atorvastatin. Looking at the overall survival curves, we see that the median OS was not reached in the statin group. The mean survival in the statin group was 225 months compared to 163 months in the non-statin group. And the difference in the overall survival between the two groups was not statistically significant, with a P of 0.29. The median follow-up time for the entire cohort was 37.2 months. When we stratified the overall survival by tumor site, the most common being the liver, the lung, and the soft tissue, the sample sizes were too small to make any meaningful interpretation. We do, however, note that in the liver, no events of death were recorded among those who received statins of the 8 patients. To summarize the findings of the study, these findings do indicate a trend towards improved survival for patients with EHE who have received statins over the course of their disease. But the study was significantly limited by a small sample size in the statin group. Looking at future directions for the study, we are looking at conducting a bigger study with a larger sample size for the same population, as well as collecting additional data on their therapies that they have received, and look at its interaction with statins and its association with survival. However, prospective studies to assess the therapeutic benefits of statins in EHE is definitely required. We also need to assess the clinical benefit of combination of statins to the current systemic therapy regimens and targeted agents that are used in EHE. And discovering biomarkers that can identify responders to statins would be very beneficial in this cohort. Focusing on these areas would definitely help in adding statins as a promising new option in the management of EHE. I would like to thank Dr. Vinod Ravi, my mentor at MD Anderson, the faculty and the study team of Sarcoma Medical Oncology, and the patients and their families for their support and encouragement to conduct this study. Thank you.
Thank you much. If you don't mind sticking around for a short Q&A. With this particular mechanism and using statins, is there anything that would be predictive at this point in terms of what statins might work better or not for this particular indication relative to why they're primarily being used?
Yes, there have been no studies in EHE, but studies in the past have shown that the lipophilic statins like atorvastatin, simvastatin have shown the most benefit in working as an anti-cancer agent compared to hydrophilic statins. So the atorva and simva have shown benefit, but those have been in cancer cell lines. Definitely need some human studies to be done in prospective studies in order to see which would actually work in humans for those.
Yeah, absolutely. And then with knowing that there might be some subcategories of statins that are going to perform better, and the answer may be that you don't have enough data points to do this, but have you analyzed your data at all looking at that subset of statins relative to the others to see if that pushed up the statistics?
Yes, the one difficulty has been that we have a really small sample size when we stratify it based on the statins, because simvastatin was only 4 patients in our cohort, whereas atorva was 14 patients, still a smaller subset. So it would be hard. So it would make more sense to look at them collectively currently rather than substratify it, yes. Because splitting them across for both the statin as well as the non-statin group would diminish our data set significantly.
Always one of the challenges with rare cancers.
Yes.
Got another question that came in from the audience. Do you know if statins shift TAZ-CAMTA1 localization?
Statins have been known to shift TAZ-CAMTA1 localization in cancer cell lines, again, in breast cancer and in multiple myeloma, I believe. But there has been some data on that. There is a paper in 2019 or 2018 that came out that did the experiment in breast cancer cell lines. And it demonstrated that TAZ-CAMTA1 localization was shifted with statins.
Excellent. That hits all of the questions in the queue. Thank you so much for your time today. At this point, we are going on a break. It was scheduled for 10 minutes, but we're going to make it a 5-minute break.